Okay, this is the second lecture on cell membranes. In this lecture, we're going to focus on active transport, or moving molecules across the membrane and that requires energy. So active transport, we're going against the concentration gradient. All of the transports that we've seen so far are going down the concentration gradient or with the concentration gradient. And this is going to require a pump because we're going against the concentration gradient. And or an electrical gradient. We're going to see how cells also use electrical gradients. So if there's more positively charged ions outside of the cell, they'd have a tendency to flow into the cell. But we can pump ions against their electrical gradient as well, and that requires active transport. And so these types of transport is going to require energy, and generally that is found in the form of ATP, which we've already met. So active transport requires proteins embedded in the cell membrane, similar to facilitated diffusion, but in this case, we're going to be going against the concentration gradient. So I don't think you need to memorize these different types, but just understanding because we're going to be talking about these different types. You don't just need to know the names. But uniporter is just going to be one directional, so one molecule moving against the concentration gradient being pumped into or out of the cell in one direction. Symporters where one molecule is being pumped and another molecule kind of comes along for the ride. So they're being like simultaneously pumped through. And then an antiporter, we'll see this as well, and this is where one molecule will be pumped in, while another molecule will be moving out. So it's going in opposite directions, so that's an antiporter. And we're going to see examples of each of these as we progress through the school year. Okay, primary active transport is just the pumping of a molecule against the concentration gradient or against the electrical gradient or both, and that is using ATP directly. So it's hydrolyzing ATP and using that energy to pump molecules against the concentration gradient. And basically what the cell is doing is it's setting up, it has now potential energy. Once we have a concentration gradient or an electrical gradient, now it has the potential to do something, because now these molecules will run down there, passively run down their concentration gradient or electrical gradient, and something can happen inside of the cell. Secondary active transport is where we use ATP to pump molecules against the concentration gradient, and then we allow those ions to move down and we use that to do something. So it may be a way of taking in amino acids or sugars, and these are going to be pumps that are going to be like symporters or antiporters are going to be used in secondary active transport. Okay, so those are pumps, moving things against the concentration gradient. Another type of transport that requires energy is when we want to move molecules that are very large. When they're too large to cross through the phospholipid layer, too large to move through those proteins, they can be taken in through a process called endocytosis. So endo means in, cyto, cell, so into the cell. And so here we have this large molecule, or large substance, whatever it happens to be, and the membrane kind of will invaginate like this, and eventually form like surround the substance, and then pinch off, and now we have a vesicle. So this vesicle contains whatever that large substance was brought into the cell. There's two different types of endocytosis. If the cell is taking in a solid, we call that phagocytosis. Our white blood cells do this in engulfing bacteria. And then there's also pinocytosis, and this is where you're bringing in liquids. These vesicles would be much smaller than what we see in phagocytosis. There's another type of endocytosis, and that is receptor-mediated endocytosis. So if the cell is trying to bring in something that's in the extracellular matrix, but maybe not in high concentrations. For example, cholesterol. We don't want cholesterol in our fluids in high concentrations, but we need to bring it into the cell. So in that kind of case, what we end up seeing is the cell membrane will have proteins on it that are specific to whatever that molecule is. So these proteins will bind specifically to that molecule. And so we're going to get a concentration of them in this area. And then again, this area of the cell invaginates. We call it coated pit just because it looks like, has a coating on it from these proteins binding to whatever substance they're binding to. And then it's again going to pinch off, and now you have a higher concentration in that area than you would have if you just randomly, invaginated and took in some substance that was in the extracellular matrix. Okay, the opposite of that is exocytosis, and this is where something is expelled from the cell. And so this is really just the opposite of this. But this is how neurotransmitters, we're going to be seeing this in our next unit, are released. So we have a vesicle, it fuses with the cell membrane and opens up, and then releases its contents into the extracellular matrix. Okay, so before we leave membranes, I just want to talk a little bit about some of the other functions of membranes besides allowing things to enter or exit the cell. And we're going to be seeing this as we move through the year. So some membranes are electrically excitable, meaning that a change in electrical charge across a membrane can cause things like nerve impulses to be conducted from one area of the body to the other, or muscle cells to contract. And so we're going to see how membranes are involved in that. Some membranes are used in transforming energy, and we're going to see this in our energetics unit. So the inner membrane of the mitochondria and the thylakoid membrane of the chloroplast. So we use these membranes to create a concentration gradient. We pump protons, use energy to pump protons, and then when the protons run down their concentration gradient, that energy is used to phosphorylate ADP to generate ATP, which is the energy for the body, for the cell. Another function that we see with these proteins is that they can organize chemical reactions. So in this case here we have the membrane proteins, and they're lined up in such a way that when we have, again, remember we talked about these series of chemical reactions that happen in our body. So if this is the first enzyme in the reaction, this is the second enzyme in the reaction, let's say, they're lined up on the membrane. And so in space, A goes to B, and now the enzyme that's going to catalyze the reaction for B going to C is lined up, and that's going to facilitate that process. And I think this is the last one. We'll be looking at this in our next unit, and some of these proteins that are bound in the membrane kind of act as, they'll kind of start a certain process, or a chemical signaling process. So this molecule binds to this protein spans the membrane, changes shape, and so now on the inside it either activates or will activate something else. So this change in shape will make something happen inside of the cell. And that's a signal started in the signal transduction pathway, which we'll spend our next unit on. So I hope this helps.